I invite you to Bologna. You can see the two towers, and the city is not as beautiful as large as Moscow, but is very beautiful. This is again some of the people who did the job. And I would like to tell you that what I will present you is a part of a study on nutrition, aging, and longevity, with particular emphasis on gut microbiota and metabolomic, in collaboration with the group of Patrizia Brigiti, Willem de Vos, and people in the Serge Rezzi and Sebastiano Collino at Nestec in Lausanne. The basic assumption of my talk is that nutritional reprogramming of the aging process is apparently possible in humans and should be pursued. In particular, we propose that the gut microbiota can be a good target for life extension, not only in humans, but also in animal models. And the other assumption that I will follow is that an appropriate diet in the elderly can reduce inflammaging by changing the gut microbiota. And the other point that is related to my presentation is that nutrition can have epigenetic effect. And I would like to remind you, among many papers, this one which has been recently published in Cells, in which they showed that these changes of a diet in male mice can be transmitted to the offspring. And indeed, the offspring of males fed a low-protein diet exhibited elevated hepatic expression of many genes involved in lipid and cholesterol biosynthesis, and decreased the level of cholesterol esters relative to the offspring of males fed a controlled diet. And what is important is that epigenomic profiling of offspring livers revealed numerous modest to 20% changes in cytosine methylation depending on paternal diet. So this suggests that something can be transmitted from the father to the offspring, and that this poses a lot of questions. So the one basic question, if the effect of diet on epigenetics can be mediated, at least in part, by the gut microbiota. We will address most of this problem in a recent approved project from European Union called New Age, is in the KBBE on a diet. The title is New Dietary Strategies Addressing the Specific Need of the Elderly Population for Healthy Aging in Europe. It started 1st of May last year, and it will be for 5 years. These are some of the partners. There are the research institutions, stakeholders, and SMEs, and large companies and enterprises. And the basic hypothesis is that an appropriate diet, an ad hoc fortified Mediterranean diet, can decrease the level of the chronic subclinical low-grade inflammatory process that I have proposed to call inflammaging. And we will recruit 1,250 individuals in 5 European countries, and they will follow a diet. Half of them will follow this Mediterranean diet, ad hoc fortified for the elderly, and half will continue their own diet. And at time 0 and after 12 months, we will measure a lot of parameters: immunological, inflammatory, genetics, body composition, and so on. In particular, in a subset of individuals, we will do in-depth immunology, transcriptomics, oxolipidomics, metabolomics, gut microbiota, and epigenetic studies at the whole genome. the idea is that the final aim is to collect clinical and nutritional data plus all this omic data and to arrive to a predictive model for inflammaging-based nutrition in the elderly. Inflammaging, this is the original paper where I proposed this idea of inflammation and aging. I think that the subtitle is extremely important, is an evolutionary perspective of immunosenescence. So it's an evolutionary based hypothesis. We still do not know much about the stimuli which the sources of this inflammatory stimuli. There are for sure internal stimuli like a genetic predisposition. You heard from Judy Campisi that the senescent cells have a pro-inflammatory phenotype. Of course, adipose tissue is a great source, but take into account that most of the aged people are lean, and so there must be an enormous reshaping of the adipose tissue all along the body, and we have several types of adipose tissue. We don't know much about their difference between the subcutaneous, the visceral, but also the mediasthenic, the epirenal, and so on. So there is still a lot of knowledge which is lacking about the fat tissue, which has been largely neglected, I think. The DNA damage response, of course, is another one, and another possible source is the circulating mitochondrial DNA, and we have data suggesting that this circulating mitochondrial DNA is increasing with age. It is recognized as a foreign bacteria product because originally this mitochondrial DNA comes from bacteria originally, and it is probably another internal stimuli, but there are probably many others. These are some of the genes and polymorphisms that we have studied in my laboratory, looking at the predisposition to produce more or less inflammatory product. I have no time to go into details. The other point that I would like to stress is that there are many external stimuli, and in particular persistent chronic subclinical infections. They can be viral, like the human cytomegalovirus, EB virus, and other viruses, and bacterial. You know that there is a large literature regarding the importance of infectious foci in the oral cavity, and I will talk about the gut microbiota, and there are, of course, other microbiota in other parts of the body. Inflammaging, we have the idea that inflammaging is a systemic condition. The gut microbiota can play a role, but the liver, the immune system, adipose tissue, muscle, and brain, to mention only a few, are probably involved directly or indirectly in this inflammation, which is both local and systemic. What about the role of the gut microbiome? The intestinal microbiota consists of an heterogeneous and largely non-culturable bacteria that orchestrate homeostasis by communicating with the epithelium and innate and adaptive immune mechanism of the gut. So it's very important to pose the gut microbiota in its situation, which is very important for the gut and for the immune system. With a density of 10 to the 12 organisms per gram of luminal content, diversified in more than 1,000 bacterial species, the human gut microbiota is indubitably the biggest, the most diverse, specific, and temporary stable symbiont microbial community of our body. But, and of course, the physiology is quite complex. There are incoming invaders, there is food, there is a quorum sensing mechanism. In green is the mucus, which is very complex and plays an enormous role. So you can look at the review of Weissenbach that has been recently published in 2011. This is another paper published in Science, a review suggesting how many are our invisible partners, the bacteria in the different body sites, not only in the gut, which is the most important here, the gut, but also the mouth, the esophagus, and so on, and the vagina, for example. So all our body has a lot of invisible partners. Regarding the human microbiota, we can say that we are 90% bacteria because they are about 10 to the 14, and our bacterial counterpart provides essential features that we have not evolved. Enhancement of digestive efficiency, competitive barrier against invasion of pathogens, development, education, and function of the immune system. And I remind you that the mucosal immune system is a great part of our immune system. Strengthening of the gastrointestinal epithelium impermeability, detoxification of xenobiotics, synthesis of vitamins, and also recently a gut-brain axis is emerging. So each human's human being has a metagenome. It's the human genome plus the collective genome of the microbiota, which is called the microbiome, and has to be considered a meta-organism. This is a very important and key point, resulting of millennia of coevolution with an incredible number of symbiont microbial cells living in our body. So humans are meta-organisms. And the dietary changes in the Neolithic agricultural society due to the invention of agriculture has favored a reshaping of our gut microbiota, and symbiont intestinal microorganism provided also probably a strategic fitness for against for protecting the host from the new pathogens that we were exposed to after the invention of the agriculture. The dietary changes, the gut microbiota so has a long evolution. There was a great change about 10,000 years ago, but is still evolving because the type of diet that we are following in our societies is reducing the biodiversity, and so there is changes in our gut microbiota are occurring also at present. Today, the widespread use of antibiotics, cesarean section, clean water, and so on, can also have a major role in reshaping the number and the types of these microbial species that are part of our body. Phylogenetic diversity. It is interesting to remember that among the many species that are available, bacterial species, only few have colonized our gut, 6 out of about 100, and these are the major phyla: Firmicutes, Bacteroidetes, Actinobacteria, Proteo, Fusobacteria, and also others. So there were specific type of bacteria that were allowed to colonize our gut. And it's important to remind that each of us has a specific fingerprint. 70% of the species phenotypes are subject-specific. No phenotype is present at more than 0.5% in all subjects. So the individual microbiota show an astonishing level of inter-individual variability. And there is, each of us has a microbial fingerprint. The microbiome is probably constituted of about 3 million genes, so about more than 100 times our own genome. And it's important to remind that only about half is quite well known, and about half is still unknown, and this is called the biological dark matter of the human microbiome. Each subject possesses one constant and one variable microbiome. This is 75%, about 75% is constant, 25% is variable. And so this, this individual metagenome diversity provides an expanded and dynamic view of the human interpersonal genetic variation. The microbial makeup can be changed by diet, the environment of the gastrointestinal tract, if there are inflammation or not, environmental factors such as microbes geography, climate, sanitation, and of course host genetics. It's also important that this gut microbiota can change quite rapidly according to the diet, the type of ingested fermentable carbohydrates, protein, carbohydrate, or fat. And dietary changes can occur and reverse in just 1 or 3 weeks. And there are people who are short-term and long-term responders. So in a general sense, the microbiota can be regarded as an epigenetic factor that, shaped by the environment, confers adaptability and resilience to the human meta-organism. So the idea is that the gut microbiota has a pervasive biological effect, as I told you. And these are the major diseases which today are affected by changes in gut microbiota. Inflammatory bowel diseases, of course, obesity, diabetes type 1 and 2, cardiovascular diseases, allergy, colorectal cancer, and what is very important is gut-brain axis, and the gut microbiota has been shown to play a role, for example, in autism and depression. Of course, chronic inflammation can have a dramatic impact on the gut microbiota, especially chronic inflammation in the gut. I have no time to go into many details. What is very important, I would like to stress the importance of the gut-brain axis, and it has been shown that in mice, but just by changing the gut microbiota, you can change the memory, behavior, so very important characteristics. So this probably gut-brain axis has been underestimated even by people who are by the neurobiologists. And it is interesting that, for example, in model system of autoimmune diseases of the brain in mice, if you change their diet and if you add a few bacteria, good bacteria to the diet, the inflammation it has been shown that the inflammation in the brain is definitely highly decreased and probably stopped. So there is a very important effect of the gut microbiota on the brain. You can study the gut microbiota diversity with several techniques: FISH, TRLFP, real-time PCR, DGGE, and phylogenetic microarrays. In this case, these are very good. We use this one. They they are unable to detect less abundant or unknown bacteria, but are quite interesting. Of course, nowadays it's possible to use Illumina platform for the complete sequencing of the entire microbiome and to make also metatranscriptomic. So in general, these cartoons should be very clear. You have a microbial community. You can ask which microbes are there, what are they doing, what is the genetic potential. And I would like to stress that you can take DNA and RNA and make metatranscriptomic. And what we are doing in particular is to look at the urine to check for metabolites which can be in the urine, which can be related to the function and the composition of the gut microbiota. We used for our studies this 16 sRNA ribotyping, which is called the human intestinal chip, the IT chip. Is based on the fact that the small subunit ribosomal RNA is present in all organisms and includes about 1,500 base pair of sufficient size for comparative sequence analysis, which remain relatively concerned in evolution. Contains hypervariable regions which have been sequenced, and so you can put in this arrays more than about 5,000 probes, which allows you to identify 1,140 microbial phenotypes. This is the paper that we published a couple of years ago, in which I think is the first paper that studies the gut microbiota in very old people. And we studied 20, 21, 22, about 20 young people, 20 centenarians, 20 offspring of centenarians, and a group of controls of this offspring. The gut microbiota in centenarians and their offspring is quite different, so there is not much relationship. So we put in the subsequent slides all the people of 70 offspring of centenarians and offspring of long-living parents were put together. This is an important result because it suggests that at age 70, what is important is the environment more than the family or the genetics. And this is contrary to what you can see in babies, because in babies the gut microbiota is quite similar to that of their mother, but probably going on with age, the diet and the environment is more important than the familiar or genetic components. This is the heat chip. Here you should see the name of the bacteria, but what you can see probably better is that 15 centenarians out of 21 were clearly separated from the others, suggesting that they have a specific and unique type of microbiota. And this is very important because it's a major characteristic of age-related changes of the microbiota, and you can see that from young to people of 70 to the centenarians, there is a decrease of biodiversity. So the number of species decrease with age. And this is a major characteristic of age, but also of age-related diseases in which the microbiota can play a role. These are some of the most important bacterial groups that contribute to the difference between these three age groups. The centenarians are here, the young are here, and the old are here, and this is the list of some of the most important bacteria which distinguish them. And this is a summary of the phylogroups which contribute to these GM profiles. And you can see that major changes occur in Clostridium cluster 14 and in bacilli, which increases with age, and in proteobacteria here. So, and what is important for us is that the redundant analysis suggests that about 9% of the total variability of the gut microbiota is related to the pro-inflammatory cytokine pattern, because of course all these people have been fully characterized immunologically, and you can see that IL-6 and IL-8 are very important, are much higher in centenarians than in these controls, and these are some of the bacteria which are related to this increase of IL-6 and IL-8, suggesting that the gut microbiota can play a role in inflammation and inflammaging. So just to summarize, the fecal microbiota of centenarians show an organization which significantly differs from the adult-like pattern. Is a highly compromised ecosystem, low diversity, decrease of good bacteria like Clostridium cluster 14, which is a butyrate producer, decrease of bifidobacteria, which are also good bacteria, and increase of facultative anaerobes, including bacilli and proteobacteria, which are considered pathobionts. So in general, our data suggests that the gut microbiota is relatively stable, even if there is a loss of biodiversity throughout adulthood, and its stability may last longer than expected, and that major changes in the gut microbiota do not follow a linear relation with age, because increase the change in microbiota in the last decades. And that an clearly aged GM gut microbiota occurs likely after age 70. GM composition of parents and offspring show no significant similarity, apart a few cases and minor similarities. Geographical variation, country, ethnicity, diet, impact on GM, and there are papers which has been published after our paper that suggests that indeed, for example, in other populations like Irish, the gut microbiota of Irish elderly can be a little bit different from the one from Italian. GM changes in centenarians are accompanied by an increase in pro-inflammatory cytokines, supporting the hypothesis that it could contribute to inflammaging. This suggests this is a theoretical summary, suggesting that is remarkable that all the changes that have been described in the gut microbiota, for example, in obesity, in diabetic type 2, in aging, and so on, are quite similar. So when the gut microbiota change in a stereotype way, and particularly is important the decrease of biodiversity. This should summarize the major changes which occur in gut microbiota during during our life. You, at the beginning, you are in the uterus, you are sterile. Immediately after birth, you are exposed to the microbes, especially in the vagina of the mothers, if you have a natural delivery. But this is not the case with cesarean delivery, which, of course, is very different from this point of view. And then there is an expansion, and so you have an infant gut ecosystem, an adult gut ecosystem, and aged gut ecosystem. This should suggest that if you are healthy until 70, you can maintain a good microbiota. But if you have any disease, major changes, the reduction of biodiversity and increase of pathogens can occur much earlier. This, of course, depends largely on the diet, which is basically liquid, then solid, and in the aged people there is a change of diet, change of lifestyle, reduced intestinal mobility, increased intestinal permeability, and of course immunosenescence. So aging is characterized by a change in the microbiota. This can be the major variables. There is a reduction in the immunomodulatory groups and probably a pro-inflammatory consortium of bacteria. We conceptualized and reviewed this topic in this review article and this other review articles, which is quite large and is a real comprehensive review of existing literature. And in this one, in this paper here, we address the role of the changes in gut microbiota in colon cancer. In colon cancer, one of the major points is that there are bacteria which can secrete mutagenic substances. And this bacteria can play a major role in the development of colon cancer. And of course, the diet and other major factor, age and so on, is important for the pathogenesis of colon cancer. And the gut microbiota likely play a major role. There are many open questions. How do nationality or ethnicity related features such as diet, environment, lifestyle impact the aging of the intestinal microbiota? How does the individual physiological health status determine the threshold of aging of the gut microbiota? Can we find specific microbial metabolites, biomarkers of the aging process? And this is what we are doing with the metabolomic studies in urine. How is possible to modulate the aging process of the intestinal microbiota? This has been addressed 100 years ago by Ilya Mechnikov, who was the pioneer of these studies, and he deserves here a mention because it was not only one of the greatest immunologists of the world, but also he touched this important point that the aging process could start in our intestine, and that from the intestine signals could arrive to the entire body. And he started the idea of using yogurt, taking these ideas from what has been used in several populations, probably in Eastern Europe, and this was the starting of the French industry of yogurt, like Danone and other companies, you know. And which is the right intervention time for the modulation of the aging process of the human microbiota. Let me say a few words for those who are interested in C. elegans and other model systems. As far as we know, in the review article that in biogerontology, we addressed all the literature regarding the possible papers which suggest that the extension of lifespan is possible not only in humans, but also in model systems, and we found that the literature is very scanty. I will mention in this case 2 papers that have directly investigated the effect on C. elegans longevity and resistance to infections when feeding with lactobacilli and bifidobacteria, so 2 types of good bacteria, the most common from non-pathogenic microbiota. They fed worms for 3 days on regular E. coli and then transferred animals on place containing this type of bacteria, and all animals shifted on the new diet had significantly extended lifespan and higher resistance to infections with pathogenic bacteria. In Drosophila, there are not so many studies. No, there are more studies, sorry, examining the relationship between microbes, gut, and aging. And here also Regina told us a lot about the integrity of the intestine in Drosophila with age. So this is a very hot topic. But as a whole, these studies demonstrate that during natural aging, the gut of Drosophila undergoes dramatic age-related deterioration of gut homeostasis that appears to be associated with shift in gut microbiota composition and density, and directly linked to the presence of gut microbiota, as germ-free flies do not exhibit this age-related phenotype. So, but this is not my field, so do not ask me more because this is not my field. I just reviewed the literature in the review that I showed you. So this is the end. I think that we can end with this, with what has been written by Bremer recently, You Are What You Eat. This was something that the French materialist liked so much during the Revolution, and this is the materialistic philosophers. And, but with the new data, we can say that you are what your parents ate. Thank you for your attention.